My name is Dan Nilsson, and I'm a professor of biology at Lund University, and I work on vision in animals, really all sorts of animals from jellyfish to man. And I'm going to give you a lecture today about the evolution of vision. The title says evolution of the eye, as if I just meant the human eye, but that's not really what I mean. I mean the evolution of eyes in general.
So good evening, everyone, and welcome to the third of the 2019 Darwin College lectures on the theme of vision. Last week, if you were here, you have heard Anja Herbert speak on vision and colour, and she showed how subjective our interpretation of colour can be, that what we think we see may or may not be correct or even there, and how many factors can affect how we visualize colour. So, eyes. The creationist's question is, of what use is half an eye? And Charles Darwin thought about that, of course. Now, eyes are wonderful things. In my own research, I've worked on the mid-ocean ridges, where the tectonic plates diverge. Many of the life forms along these deep-sea ridges are endemic and only found in those very extreme chemical, temperature and pressure environments, basically around the hydrothermal vents, the hot water springs there. In the Atlantic, shrimps swarm around the hydrothermal vent chimneys and feed on chemosynthetic bacteria. Now, Cindy van Dover discovered that these shrimps have evolved the most remarkable eyes. They're located on their backs and are essentially just naked retinas, adapted for detecting the radiation from the vents, this hot, very hot water. Remember, there is no sunlight 2 or 3 kilometers down in the ocean. So the topic of tonight's lecture would, I think, delight Charles Darwin. The topic is the evolution of the eye. So please welcome Professor Dan Erik Nilsson from the Department of Biology at Lund University in Sweden to speak to us.
It's a pleasure and an honor to be here and give this lecture. Actually, had there not been an evolution of the eye, you wouldn't have found your way to this lecture, and I probably wouldn't be here to lecture about it either. So it's because of the evolution of the eye that we're all here. I'll try to give you some idea of why, when, and how eyes evolved. It's been some controversy about it a long time ago, starting with Darwin, of course, but also in more recent decades. But nowadays, I would say there is not much controversy left. We pretty well understand how eyes evolved, why they evolved, and when they evolved. And that's what I'm going to talk about. So we'll try to bring more clarity to the question. Of course, the eye is not really, the eye is a complex organ. It contains many different parts, and that was the original problem, to try to understand how you can get that many parts at the same time. But you don't really need to have them at the same time, it'll turn out. We'll come to that pretty soon. But I will actually not start to try to simplify things. I'll start to make a mess instead by complicating things. Because the title was actually not really the intended title. The title says Evolution of the Eye, kind of implying that I was going to talk about human eyes. That would be just a small part of the story, so the talk is really about the evolution of eyes. And then we need to see what kind of eyes there are. We have vertebrates ourselves, and we share eyes that are very similar to ours with other mammals, with birds, reptiles, amphibians, and fish. That's actually a fish. It's as you can see on the blue spot, it's a blue spotted ray, even though it looks like a frog, but the frog is actually over there. We've got other animals. That's not a vertebrate eye. That's actually a cephalopod eye. It's a squid eye. Very similar in general design to ours, but if you look at the details, they are very different. There are spiders. That's a little jumping spider. It's got beautiful eyes. We've got insects with their compound eyes. There are many different types of compound eyes even. Crustaceans also share compound eyes, also of many different types. Do you know what kind of animal that is? Nothing you see around every day. I can see a friend of mine over there who's got a Tasmanian wife. They are from the southern hemisphere. It's a velvet worm. They've got tiny little eyes, but it's a little lens and everything is there. That's a gastropod mollusk, conch. Doesn't seem to really understand why it's got its eyes, it seems. That's a ragworm. It's got 2 pairs of eyes on its head. And that's a flatworm. It's got a pair of really, really simple eyes. But it's actually even more complex than that. This lizard has got a 3rd eye in the middle of its head. This is a bee, like other insects. They've got an extra set of, not compound eyes, but single lens eyes in the middle of their head. This is a marine flatworm. It's got a compound eye, single compound eye in the middle of the head, if they have a head. Jellyfish definitely don't have a head, but they still have eyes. These are eyes of a jellyfish, of a box jellyfish. These eyes, the little blueberries there on the scallop, that's actually eyes, a long row of eyes, but not on the head. That's actually sitting on the part of the mollusk that is called the mantle, the part that generates the shell. Now producing heaps of eyes. A relative to scallops, that's the same part, the mantle of an arc clam. There are lots of compound eyes along there. That's the tentacle of a sedentary worm, of a fan worm. On the tip of the tentacle, it's not really their normal eyes on the head. They've developed little compound eyes. That's a compound eye at the tip of the arm of a starfish. This animal has vision. Sea urchins have vision, but they haven't really got any eyes. Instead, they've got light receptors, small light-sensitive cells, sprinkled across the skin and looking through, or actually between the spines. So the whole animal works like if it was a compound eye. Chitons also have dispersed vision. Numerous little black dots here are actually little photoreceptors or eyes. So they combine the information of all these to respond to their environment. So this is a more complex picture of the eyes we know of today. All sorts of eyes in various parts on the head and basically on any other part of the body. And if you ask, could you remove any parts of these eyes and the system would still work? Most often you would know, actually, if you remove this and that, the system wouldn't work anymore. But there is one thing that you could make any use of. If you ask the question instead, what kind of thing could be useful in these eyes if you removed everything else? The only answer to that is the light-sensitive cells. You could actually make use of light-sensitive cells if you had nothing else. And lots of animals do that, because all animal eyes and light sensitivity is based on the same molecule, or the same, it's actually vitamin A, or different types of vitamin A. And that is a really clever molecule, because when it receives light, it changes conformation, and you can easily flick it back through an enzyme, or even by using light again. You can flick so you can reuse the molecule over and over again. It's a really, really useful molecule for detecting light. It sits in a receptor protein, which is called an opsin, and all animals have their vision based on opsins. In the opsin, you find the little vitamin A molecule, and the opsin is responsible for actually starting the events in the cell that leads to a nerve impulse, to an electrical impulse. But if we take a closer look at these opsins, because it turns out that there is not just one opsin that all animals share. There is a whole group of different opsins, or actually it's a whole family or class of opsins. There are many different subgroups, which will remain or return to later. C opsins you should remember, and R opsins, which are important. Then there is another group, often referred to as tetraopsins, and there is actually a handful of other groups which is not even in this diagram. One important thing we should note is that the closest relative to all these opsins is actually melatonin receptors, because opsins are part of this large family of receptor molecules that we use for taste, for olfaction, for smell, for neural transmission, and for hormone communication in the body. So it's a huge family of receptor proteins. Just a small part are the opsins. And the closest relative is melatonin receptors. You know, melatonin, that's the hormone, our sleep hormone, that tells us it's night and we should go to sleep. It turns out that melatonin itself, the molecule melatonin that the receptor is sensitive to, could actually be a light receptor. And it is a light receptor, it seems, in some single-cell organisms. Because it is light sensitive, if you shine light on melatonin, it oxidizes and it no longer fits the receptor molecule. So if you have a constant production of melatonin, and at night you will build up high levels of it, and you will be able to know it's dark. As soon as day breaks, light will reduce or actually not reduce, it'll oxidize the melatonin and make it ineffective. So that it's quite possible that that was the precursor of all visual events in all animals. But it's a kind of useless molecule anyway, because it is sensitive mainly to ultraviolet, which is, it's hard to make it sensitive into the visible. And also you kind of ruin it by shining a light on it. You have to produce new melatonin all the time. The vitamin A molecule is far better because you can change the conformation of it and reuse the same molecule. So by just modifying the protein a bit such that it works with vitamin A rather than with melatonin, that might have been an early and really good change that made all vision possible. Take a look again at this cluster of different opsin groups. The interesting thing is that these groups contain opsins from all major groups of animals. So all major groups of animals share every type of opsin, which means that our earliest ancestors, before they actually branched into the different animal groups, must have had all these types of opsins. Knowing that, we can try to turn the clock back and see when we can actually time and work out when this might have happened. If we look at another type of phylogenetic trees, namely those of animals. So these are all the animals. We've got sponges down there, jellyfish up here, insects, various kinds of mollusks and other invertebrates, starfish up there, and our relatives are up in that corner. And now this whole thing is timed, so we've got a time scale on it. And sponges, they don't have any opsins at all. All the other groups have all the different types. So then we can just follow here and see it's between roughly there from around 700 million years ago. Now this graph is actually not the newest one. So people keep changing the dating of animal phylogeny. So these things would now be closer to 800 million years ago than to 700 million years ago. I don't know about these things, but I trust people that make these dated trees. So that's a long time ago then that opsins appeared. So Opsins must have appeared after that time, and all animals would have acquired the 4 different, or possibly 5 or 6 or 7 different types of classes of Opsins shortly after. So we know roughly when that happens. But that is only Opsins. What can you do if you just have an Opsin? You can express it in some sensory cells. Opsins reside in membranes. All these receptor proteins are membrane bound. So if you pack lots of Opsins in your membrane, you can make a cell that is sensitive to light. And so what might you have such a thing for? What could it tell you? It'll inform you about the ambient intensity, which tells you if it's night or day, of course. You can use it for knowing about lunar rhythms or yearly rhythms. You can actually also use it for UV, warning for too high levels of ultraviolet radiation, which is harmful. You could use it as a shade detector. You could use it as a depth gauge, which might have been a really important reason for these early Opsins. The deeper you are in the sea, the less light there is, so you could regulate your depth if you just measure light. If you're living in sand or gravel or in the mud in the seafloor, if you're burrowing there, you could use it to determine when you actually break the surface or are close to breaking the surface. So there are a number of different uses where you can just, where a cell that is just sensitive to light from all directions is actually useful. So it must have started something like that. So we now know why and we know roughly when. There are some animals that have just that still. This is the larva of a sea urchin. It has these non-directional light receptors in several parts of its body to control circadian rhythms and control as a depth gauge as well. But we need kind of a tool. We could use an interesting tool here if we ask the question, what can a receptor discriminate? What can a light receptor tell apart? What kind of tasks can it be used for? If you want to use it for knowing that dawn starts, then you don't really need to discriminate very small intensity differences, because the difference between a starry night and a sunlit day is 8 orders of magnitude. It's a huge difference. So you don't need to be very precise. But it turns out you need to collect photons and basically count photons. Even if our eyes do it or photoreceptors do it in an analog way, they still count them. A small number means a large uncertainty. So to be able to discriminate a certain level, you need to collect a certain number of photons. And then we can make a little mathematical tool which tells us at what ambient intensity will the system work. Is there enough light for the system to discriminate what it should do? And in order to tell night from day, then it's easy. Small differences require a large number of photons, of course, but we calculate it, and then it turns out that a system like this would work down to starlight. That's 8 orders of magnitude, or 8 log units below sunlight. It will work. There is enough photons around to tell something about the ambient intensity. So we'll use this tool again with another outcome soon, because it is important. You can also ask, can we squeeze any more information out of this cell? Wouldn't it be nice to know where light comes from? That would give us completely new ways of using it. The only way to make this system sensitive to light only from some directions is to add some dark screening pigment that stops light from some directions but not from others. And then we'll get another completely different function. We'll get a directional light sensitivity, which informs about the direction of light. It can be used for phototaxis to guide animals toward darker places or towards brighter places. It can also be used by the animals to control their body posture, which might be useful to know what is up and down, because light usually comes from above. But there, of course, might be a little problem. So let's apply our tool again to this system and ask, at what intensities would this thing be able to? Because now we're not interested in discriminating the huge differences between night and day. We're interested in discriminating brighter part of one scene from a dimmer part of the same scene at the same time of the day. And the differences are now much smaller. Plus, we have cut off half the light. And another thing, the changes over the day are slow, so we can integrate over a long time. Now we can't integrate over such a long time anymore. We have to make a much shorter shutter speed, or a higher shutter speed. And all those things means that we need much more light. So if we do the same calculation, it turns out that this thing will work to about 4 orders of magnitude below bright sunlight. At bright sunlight during the day on land. If you're in water, let's just about 3 meters of depth will remove 1 order of magnitude. So at 6 meters, 2 orders of magnitude are gone. At 9 meters, you've lost 3. If it's an overcast day, you've lost an additional 2, which means that you easily eat up those 4 log units, and then the system will not work anymore. So this system is actually not really very good, because under very rare conditions will that cell with just opsin in its membrane be sensitive enough to do the discriminations. You really need more sensitivity. One of the problems is that it's only about 0.02% of the light that is absorbed by 2 passages through the membrane. But it's, of course, easily solved. Just fold the membrane, so light has to pass through many more layers of membrane. So you make a stack of membrane instead. You can make a much bigger stack than I've illustrated here. And then sensitivity goes up, and the thing starts to work again. And you can now easily make it work down to 7 orders of magnitude less than bright daylight, which is perfectly okay. Then it'll work at dawn and dusk comfortably. So now we've found something that is interesting, stacked membranes. And does that exist in light receptors? Yes, it does. This is a case of all these animals we looked at before. This is the larva of a ragworm. It's got 2 directional light receptors like that. That's a flatworm with 2 light detectors like that. That's a jellyfish larva. In all these cases, there is a little pigment cell. Actually, in that case, it's the same cell that does everything. There's a pigment cell, and there is a sensory cell, and the sensory cell has really massively folded membrane. Interestingly, there are just 2 ways of incorporating opsins into stacked membranes. There are just 2 ways of stacking membrane, or making lots of membranes in a small volume. One way is to making microvilli. You use actin filaments to push out the membrane. The other way is to use cilia, which use microtubules instead for the same purpose. So modified cilia is one way of making lots of membrane. Microvilli is another. You remember we said when we looked at the opsin tree, I said that there were C opsins and R opsins, 2 different types of opsins. It turns out that there is a particular class of opsins that are associated with microvilli, and there is another type of opsins, the C opsins, that are always associated with a modified cilia throughout the animal kingdom. Which means that these events this innovation of folding membrane has probably happened twice. Really early in animal life, it has happened twice. And it has happened twice in the same common ancestor of all animals as well, because actually all animals have these 2 types, but they use them in different types of light-sensitive organs. There is another way to increase sensitivity, and that is just to have more of these cells. One cell has a particular sensitivity. If you add more cells, you have more sensitivity. And if you have a number of cells, you could sum their output in the nervous system, but you could also compare it, because these cells would actually shield each other a bit. And if you make any little change, like change the curvature like that, these different cells will have very different views of the world. They will look out in different directions. And suddenly, now we actually have the first eye. This is the first vision. Everything we've talked about before is not really vision. It's light sensitivity, non-directional light sensitivity or directional light sensitivity. But this is vision. Even if you were only to have 3 different cells, it would be an image of 3 pixels. It is still vision. And this is an example of an animal that has something very similar to what we modeled there. This is the eye of a flatworm. It's a single pigmented cell in which you find a number of sensory cells, light-sensitive cells with folded membrane. Other animals that have this, which we can now call low-resolution vision because it's not really very sharp anymore, and in these cases, it's actually not sharp enough to see other animals. They can see other animals when the other animals are so close that you can basically smell them or touch them. So it's not a long-range sense in that respect. But they can see their inanimate world. They can see big structures in the world around them. They can use it to select their habitat. That's basically what animals use it for, that have just this low-resolution vision. They can work out that it's a good place is over there and a bad place is over there. They know where to go. And they can use it, of course, for even better body posture control, and they can use it for movement control. It'll tell them how much they move in relation to the environment when they turn. They can navigate in straight lines and so on with this kind of vision. So it's pretty useful, but they cannot use it for seeing other animals. They can't see predators, they can't use it for seeing other animals to prey on them, and they can't see each other, or the same species. No, actually, I should go back here because, yeah, we've got different animals that have this is, it's a jellyfish, and we've got the ragworm that we talked about before, flatworms, and here we have a starfish which have got a compound eye at the tip of its arm. But wait a second, we haven't talked about compound eyes. How did that turn in? How did compound eyes evolve? Let's go back a bit to this number of receptor cells with screening pigment next to one another. If you turn it the other way around, not fold it up like that, but twist it the other way around, you will get the same kind of information into it as when you got a pigment cup. But now this is the start of a compound eye instead. So there are two actually equivalent solutions to the same problem. That's another way of getting exactly the same information. So now we've worked out why there are both single-chambered eyes and compound eyes. No difference in the beginning. Later on, it turns out that single-chambered eyes is actually a better solution. Compound eyes is not the world's most efficient way of using the space you've got to your disposal. So let's go back to our single cup eye like this, and we can ask the question, how would you improve it to get better resolution? Well, you actually have, of course, to squeeze more of these light-sensitive cells into a larger cup. Then each of the cells would get a light from a narrower angle in space. If you have a narrow angle in space, it means that when you move, things will happen faster. So you would have to speed up your process of vision. Your integration time or your shutter speed would have to be faster. So you integrate over shorter time, which means you collect fewer photons for each of these moments. And there is also a narrower angle. So if we apply this tool that we tried before to calculate what kind of intensity, ambient intensity you would need to make it work, then it turns out that this only works down to a couple of orders of magnitude below sunlight, so it's practically useless. Looks good, but it will really never work. If you have a very small number of cells like the flatworm, it will work on sunny days. But no, this will, and actually you don't find any eyes like that really in the animal kingdom that looks like that. One way of solving it is actually to actually really exploit this membrane stacking, to make the stacks really huge. You have a problem here, there's an open space there, that would probably very rarely happen. It's a bad thing to have an open space like that. It'll collect lots of crud in there that you have to look through, or some other smaller organisms may find it a cozy place to live. So you better put some cells in there to stop that from happening. If you've got transparent cells in there, you could just increase their protein concentration, especially in the center, and you would actually get a little lens. A lens that converges light. So you got a concentration of light from one point in space to a smaller area here, and that would increase sensitivity. So if you do these things, you extend the stacks to as long as you, so you almost absorb all of the light, and you put a little lens. It doesn't need to be perfectly focused on the retina or anything. You increase your sensitivity anyway, and the result is that that will actually work deep into dusk and dawn. So it works again when you've made these improvements on it. Are there any animals now that have eyes that look exactly like this? Well, kind of almost. That's the eye of a box jellyfish. Comes very close to what we modeled here. There are more examples. A ragworm has an eye that is almost identical to what we modeled here. There are actually yet other ones. These ones have eyes that come pretty close as well, that type of eye. So what do you see with such an eye? Can it provide any useful information? We can actually model that by using ray tracing techniques. So we work out computationally what each individual receptor cell will pick up from the environment, and we can take normal images with a camera, we can process the images and see this is what the velvet worm would see. That's the result, that's what we see. That's taken with a fisheye lens, so it has 180 degrees field of view. That's the field of view, total field of view of a velvet worm with the velvet worm resolution. Now that looks useless. If a human had that kind of resolution, you would probably call that person legally blind. You wouldn't be able to drive a car. It would be allowed to, I guess. Velvet worms don't drive cars anyway. But it's pretty useful for them anyway, because what they need to know is where dark structures are, so they can hide under them at dawn when they have been out hunting at night and the light of dawn starts. They need to find a shelter, otherwise they would dry out and die. Also in the evening, they would have to work out what direction to get out of the shelter and into their hunting grounds. So that's good enough for a velvet worm. It's their purposes. They can select their habitat and they can be in the right place, but they don't see each other until they actually touch them. This is a box jellyfish. It's got, you see one little eye up there. They've got 4 of these little clusters. Each cluster looks like that. It's got a couple of eyes. What do they see? Now that's what a box jellyfish would see if they went on a roller coaster ride. Now you've always, all of you have thought about that for all your life. What does the box jellyfish actually see when they go on a roller coaster ride? Now you get the answer. It's not all that much. It's good enough, though, for box jellyfish. This particularly the species here, it lives in mangrove swamps. It needs to steer around mangrove roots because there are lots of nasty organisms on the roots, so they don't want to collide with roots. They never get stranded because they can see when it's too shallow. They never drift out into the mangrove lagoon because they want to stay under the mangrove canopy. That's where there is lots of food. They don't see their food, but they actually see the mangrove canopy. So they can, that resolution is good enough to steer away from mangrove roots and to bring them back in under the canopy. It keeps them in the right habitat, but they don't see each other. They don't see the food. Now that may not be really the most natural habitat of box jellyfish, but it's kind of fun. I'm not sure the box jellyfish would find it that sort of amusing. I'm not sure they would even realize what's going on. That's another matter. Maybe they would get motion sick. So if we want to, now we've talked about low resolution vision. There are lots of animals that have low resolution vision. They can't, and the definition of it is that the resolution is not good enough to see other animals. So they can see the inanimate world, and they can use that to navigate and move within the inanimate world. All these eyes are small. They are really less than a millimetre. So if we want to improve on this system, we have to make something larger. And that is what has happened. The animal groups that have high resolution vision, good enough to actually see other animals, they have much larger eyes, much bigger than a millimetre. And then you can actually see things. Then you get enough light in there as well. And it's really the lens that makes it sensitive enough, because you need to concentrate light rather than having the light that reaches a naked light-sensitive cell, which may be 5 microns across. You can have light that reach a lens that is 5, 10 millimeters across, which is a huge area compared to the single cell. So the sensitivity is way bigger. And actually, animals with these types of eyes can see at any kind of place on Earth. You can even see in the deep sea at really low intensities, much lower than we can see. We'll come to that very soon. Animals that have high resolution vision, it's not actually all that common among animals. It's only a small number of animal groups that have it. Vertebrates is certainly one. We find it in insects, we find it in some spiders, and we find it in cephalopods, in octopus, squids, and cuttlefish. Those are the groups, and then there are a few little exceptions among other crustaceans, and a few little exceptions among some other mollusks and some ragworms, that isolated cases that have just made it across the border to high resolution vision. But it's mainly these large groups that have done it. There is an interesting difference then between the vertebrate eye, whether it's aquatic or terrestrial, and octopus and squid eyes. They may look identical. An octopus and squid eye may look identical to a fish eye if you look at them at low resolution. But if you look at the details, you'll find that vertebrates have ciliary-based rods and cones with Opsins in them that are turned upside down, so they actually point away from the light. That's the silly solution we have, where we actually need a blind spot to get all the axons out of the retina, or axons out of the eyeball. And in the cephalopods, it's much more wisely designed, because they're the light receptors actually facing the light. They use microvilli and not modified cilia, and they have the other type of opsin, R-opsin. So that's clearly evolved in parallel, completely independently, reached very, very similar end results. And as I said, this solution can be used for really low intensities as well. It's just a matter of making the eye really large. Some animals have made the eye really large, and those are particularly deep-sea squid. There is a colossal squid here, actually part of it. The eye is over there. That's the eyeball, and that's the opening where you can see a part of the iris there. Actually, it's frozen, and it's just thawing up. My hand is over there as a scale bar. That eye is 27 centimeters across. It's a pretty sizable eye. A relative to that colossal squid is the giant squid over here, which has also got an eye of the same size, 27 centimeters. The scale bar there is an American fuel hose, which runs right over the pupil, so we could know exactly how large the eye is. And these eyes are useful at 1,000 meters depth. They can actually see sperm whales hunting for them at that depth. The sperm whale can't, even though it's got pretty big eyes, can't see a thing. Sperm whales use sonar instead to detect the squid. So when did this high-resolution vision appear? If we go back to Cambrian fossils, it seems in the early Cambrian there were plenty of animals that had high-resolution vision. Most of them actually had compound eyes in those days, but there were also representatives with single-chambered eyes like ours. So high-resolution vision, which allowed visually guided predation, it also really called for good eyes to see predators and escape from them, and animals that interacted with vision within the species to find mates, to determine if the mate is, what kind of sex it is, to communicate visually. All those things seems to have been around about 540 million years ago. But just a little bit earlier in the Ediacaran, there were nothing like that. So high-resolution vision seems to have evolved pretty suddenly, or pretty quickly at least, at the onset of the Cambrian. So now we've timed that event as well. We know roughly when it must have happened. And we can now look at the timing. We know that that's the border between the Cambrian and the Ediacaran. So around that red line, 540 million years ago, plus minus a couple of 10 million years or so, high-resolution vision must have evolved from low-resolution vision. We can also say that the complete other events from just the first light sensitivity with Opsins to low resolution vision being pretty good, that must have happened in this almost 200 years, which may actually have been almost 300 million years if it started around 800 million years instead. So most of the eye evolution would have been completed in that time. Many years ago, I made a calculation of how many generations it would take for an eye to evolve, just to make with low selection or with weak selection, just to make the modifications in shape. And it turns out that can be done in 350 generations. If one generation is 1 year, then that's 350,000 years. That's not really a very long time on these geological scales. So eyes could have evolved many times in this period here. It's not a hard thing to do. But then again, it's not actually eyes themselves that had to evolve. Just an eye on its own is a rather useless thing, unless you have a brain behind it or a nervous system that can generate behaviors. And what has actually been driving evolution all the way is not eyes themselves, but it's the visually guided behaviors. So visually guided behaviors must kind of be there first and put some pressure on better performance of the visual organs, and then selection will actually act on the visual organs. But primarily, it works on the visually guided behaviors. So vision is really a matter of the evolution of visually guided behaviors, and I think we've now worked out what the different tasks and the different types of behaviors or in which sequence they actually evolved. It must have started with non-directional light sensitivity, which turned into directional light sensitivity, still not vision, which turned into low-resolution vision. We know roughly when that happens, but not very precisely. We know more when that happened. We know quite precisely when high-resolution vision evolved from low-resolution vision. That was at the onset of the Cambrian. But now, of course, there are things that complicate matters. There always are. So it doesn't always go this straight, simple sequence. Also, something has actually happened after the Cambrian till now. It was not that everything stopped. A number of new cases of eyes have actually evolved. These are some of them. The eyes we find in scallops and in arc clams. These are not eyes on the head. These are eyes on another part. These animals have been modified after the Cambrian explosion into other body shapes. New eyes have evolved in new places. And in this case, it seems that it must, because all clams have light receptors, which are used as shadow receptors on the mantle edge. And a small group of clams and scallops, or scallops being one of the groups, have actually evolved eyes. And and they use it for predator detection. So they're basically burglar alarms. They don't use it to see each other. They don't use it to hunt for anything. But they are still, if we use the classification that high-resolution vision is when you can actually see other animals, that is high-resolution vision. But it is maybe developed straight from light sensitivity. So bypassing the low-resolution or directional light sensitivity and low-resolution vision. We have an equivalent case in fanworms, two different groups of fanworms, the sabellids and the sapulids, seems to have done it in different ways. These sabellids, they have one pair of these feeding tentacles that are modified with a little compound eye on top. If you look into the photoreceptors, they look like nothing. They've reinvented the wheel. It is not, they haven't borrowed a photoreceptor that was somewhere else. They've really reinvented everything. They still use Opsin, though. Same thing here, they've turned or generated a boomerang-shaped eye, which is good enough as a burglar alarm. It'll tell them when a fish is approaching and about to take a bite of their tentacle crown, and then they'll see that before the fish can react. The whole worm is pulled back and it's not visible anymore to the fish. So it serves a good purpose for them. Now we can actually start to ask the question, how many times have these different transitions happened? And we now know quite roughly at least, how many times it has happened. The melatonin receptor has probably evolved only once. From that, it seems Opsins have evolved only once. From these non-directional photoreceptors into directional photoreceptors, it has happened at least 13 times, possibly twice as many times. It's hard to say, but at least that number. From directional photoreceptors to low-resolution vision, so that's the origin of real vision, has happened at least 15 times. Sometimes it turned into single-chambered eyes, sometimes it turned into compound eyes. So we have the compound eyes on the various worms and compound eyes on starfish arms, and we have the compound eyes of arthropods there. Flatworms actually also have compound eyes, some of them. And then from low-resolution vision to high-resolution vision, that has happened about 9 times, possibly more. So we know how many times all these things have happened. We know why it has happened. We know roughly how it has happened and when it has happened. So I think we now can answer all the questions we set out to answer. Why, when, and how. Why, when, and how. Yes, that's exactly the questions I think we've answered. Thank you.
So, thank you. That was just amazing. I knew that there was a great variation in eye structures, across so many animal kingdoms, but I really hadn't appreciated how deeply rooted the similarities and many of the differences were. That's just extraordinary. So, just thank you so much for coming and making all this seem so simple. And really, in reality, it's obviously so complex. Next week, we're moving away from biology. Next week's lecture, it's going to be the 4th in this series, is the vision of future technology. So Miss Sophie Hackford, who is a futurist and co-founder of the data and AI artificial intelligence company, 1751 Labs, is going to come and talk about new technologies and how interconnectedness means that our world is becoming entangled with machines. Sounds like scary stuff to me, but I hope we'll see you then. But Dan, thank you so much for coming to Cambridge to talk to us tonight. It was a delight to have you and to hear what you've got to say.
Thank you. It's my pleasure.